Good morning, friends. Today, one more important RNA virus family we will study. Its name is Rhabdoviridae. So, in today's class, we will study about the classification of Rhabdoviridae, followed by virion property, genome property, and at the last, we will see virus replication. Now, we'll start with the classification. The family Rhabdoviridae classify under order mononegavirals. Mono means single, nega means negative sense. These two things are characteristic of this particular genome of Rhabdoviridae. So genome of Rhabdoviridae is single-stranded and that has negative polarity. Virals, that is a taxonomic suffix that used to denote any viral order. So mononegavirals that comes or that has four important families which causes diseases in domesticated animal. The first Bonaviridae, Filoviridae, Paramyxoviridae and Rhabdoviridae. Now in general, if you will compare the characteristic of all these four family, the important characteristic is that is a virion morphology. So Paramyxoviridae that is pleomorphic means there is no defined shape. That is a characteristic of Paramyxoviridae viruses. Second, that is Rhabdoviridae. The viruses comes under Rhabdoviridae family that has characteristic bullet shape. Bullet shape, that is an important characteristic of rhabdoviruses and that may ask in your exam. The philo word itself tells that it's a filamentous like, or you can say thread like. That is a characteristic of virus comes under Filoviridae, and the Borna that have characteristic spherical shape, or you can say icosahedral shape. Now these are the various characteristic of family Rhabdoviridae, and we will study each and every characteristic in details in coming slide. Now this particular family contain many genus, but there are three important genus, namely Lysavirus, vesiculovirus and ephemerovirus, that contain important viruses which cause economically important disease in domesticated animal. The genus Lysavirus contain the virus rabies virus, which is responsible for causing rabies disease in domesticated animal. Genus vesiculovirus that contain vesicular stomatitis virus, it causes vesicular stomatitis disease, whereas ephemerovirus that cause disease ephemeral bovine ephemeral fever, that is also known as 3-day sickness. Now coming to the virion properties. Virion is about 100 nanometer in diameter and about 400 nanometer in length. Because of this, it gives a characteristic bullet shape. In this figure, this is an electron micrograph of rhabdovirus, which shows a characteristic bullet shape like structure, and it is so peculiar that it can be used for diagnosis of these viruses based on electron microscopy. This virus have helical symmetry and viruses are enveloped. As viruses are enveloped, they are thermolabile and sensitive to UV lights, whereas in slight alkaline environment they are stable, but they can easily destroyed by the detergent based disinfectant. Now we will move towards the genome property. These viruses have linear, negative sense, single-stranded RNA genome. This genome generally varies from 11 to 15 kilobase pair in size, and that encodes 5 important genes. These genes are interspersed with various pseudogenes. Their functions are not known at this point of time, but they may play important role. These 5 genes, which comprises of various structural and non-structural protein, and generally they are named as N, P, M, G and L. Now the location and size is depicted in this figure. This length of bar showing the size of that particular gene, and this is a location from 3' end. Now towards the 3' end, the first gene, that is N gene, that is known as nucleoprotein, and this is generally present or associated with RNA genome. Whereas second gene that stand for P, there are some books which are telling it or naming it as a NS or M2, means matrix protein 2. This phosphoprotein, this is depicted like this, and this is generally present along with RNA inside the nucleocapsid. The third, that is M, that stands for the matrix protein. Generally matrix protein, this is a location where matrix protein is present, and this helps in assembly of new virion. G, that is a important, and that is abbreviation of glycoprotein. Glycoprotein are generally present over the envelope, and they are playing important role in tissue tropism and virus neutralizing antibody production. Last, that is L, that is a non-structural protein. Generally L is stand for polymerase enzyme. Here in this case, this polymerase enzyme is RNA dependent RNA polymerase enzyme, and this is associated with virion, and that is required for their initiation of replication. Now we will see the important functions of important genes. The first gene, that is glycoprotein gene, and glycoprotein gene have two important functions because they are located on an envelope, so they are the key to decide the cells and to bind with the receptor over the host cell. So that's why they are considered as a best suitable substrate for cellular attachment. And second, they are responsible for production of virus neutralizing antibody. Virus neutralizing antibodies are those antibodies which are capable to neutralize the virus so that virus are not capable to infect further, and they are generally useful in therapeutic as well as diagnostic purpose. Nucleoprotein, that is the second most important protein. This protein has it is present on 3' end, and they are present in most abundant among all the 5 proteins. That's why they are the chief target for development of diagnostic test for rhabdoviruses. Phosphoprotein and matrix protein, these are the two proteins generally playing important role in assembly of new virion. And the last, that is L protein, that is abbreviation of RNA dependent RNA polymerase, and that helps in translation, transcription and replication of virus. Now this particular virus is of mononegavirals order that have one peculiarity, that is known as RNP complex. RNP complex means ribonucleoprotein complex. It means that the viruses comes under mononegavirals, that genomes are not present alone inside the capsid. This genome generally associated with some structural protein and non-structural protein. For example, in case of this rhabdoviruses, our genome is RNA, and that is associated with 3 important non-structural protein, namely nucleoprotein, polymerase and phosphoprotein. These are forming ribonucleoprotein complex, and that helps to stabilize and that helps to initiate the replication in newer host. Similarly, this is a photograph that's showing the RNP complex of influenza virus. Now this is a peculiarity of all the viruses belong to the order mononegavirals, and particularly this is creating problem in reverse genetic. So reverse genetic is a process for rescuing the virus from the genome. So this is a reverse to normal genetics. So here genome is not present alone, but they are always associated with some associated structural non-structural protein. So it would be difficult to rescue the negative sense RNA viruses which have RNP as their characteristic. So RNP, again, that is a characteristic, and that is helpful for stabilization of viral genome. Now moving to the last portion, that is virus replication. Virus replication we had studied in detail in general virology. Now here we will see the peculiarity of virus replication of rhabdoviruses. First, that is a entry of virus. As I had said you earlier, entry is strictly restricted based on the surface glycoprotein, or you can say they are the spike or the peplomer that can recognize the receptor over the host cell, and generally receptor for this rhabdoviruses are acetylcholine, ONCAM or some related to this one. and generally these receptors are present over the nervine systems. That's why their chief target site is nervous system, and that's why if you can see the disease caused by these viruses, they have nervine signs. The second step, that is uncoating. After entry inside the cell, they present inside the endosomal vacuoles. Now, as the pH changes, this endosomal vacuoles membrane get fused with the envelope of this virus, and that will leads to the uncoating of RNP complex in the cytoplasm of host cell. So once it will comes out from the endosomal vacuoles to the cytoplasma, now they are ready for next step, that is transcription and replication. Now transcription and replication is important step, and here you can see the arrangement of gene. Earlier also I had told you that it has 5 genes and they are present in this order, and their length are depend on the length of this box. Now 3' end, that has only one single starting point, and from this starting point all this one are translated or transcripted. Now you can see that the first step is, based on this RNA genome, they will produce the corresponding messenger RNA, and that messenger RNA ultimately translated into corresponding protein. So here they will translate it in N, P, M, G and L protein. Now you can see that this is a transcription and translation. After translation, they will goes to different place as per their function. Now coming to the second portion, that is replication of genome. As earlier I had told that this is a single stranded negative stranded RNA genome. So negative stranded RNA genome, this negative strand is act as a template, and virion associated polymerase, that is RNA dependent RNA polymerase, will help to convert anti-genome copy. Anti-genome copy is complementary to negative strand complete genome, and this is, in sense wise, it is a positive strand. Now again this positive strand act as a template, and that will use to produce the complete genome of negative strand. Now this negative strand genome copy along with various structural non-structural protein produced in the transcription and translation process, they will go to the next step, that is a assembly step. Now here we have to understand one mechanism that is a characteristic of generally rhabdoviruses, or you can say mononegavirals. Mononegavirals that have attenuated transcription, or you can say stop start transcription, or you can say stuttering transcription. All these are the synonyms. Now what it means? It means that if you can see the genome, it has a single start point, but there are multiple stop start at each juncture of the genes. So what will happen? For arbitrary example, if you will take that 100 polymerase will start for transcription at this starting point, then only 70 will bypass this junction. Means only 70 can go for transcription of P protein. So you can say if 100 copies of N protein or nucleoprotein will produce, then only 70 copy will produce for phosphoprotein. Again, out of this 70, only few, for example 50 will cross this junction, and that's why only 50 copies of matrix protein will produce. And similar fashion will happens for next two junction. So in nutshell, you can say that maximum number of protein will produce for the genes which is located at the 3' end, that is nucleoprotein, and the minimum number of gene or protein will produce for the genes which is located at 5' end, that is L, that is polymerase. That's why that will produce the gradient of these genes, where highest copy of N will produce and lowest copy of L will produce. And that's why earlier I told that N is considered as a chief target for diagnosis of this disease, because N is present in most abundant quantity. So this is a attenuation transcription, or stop transcription, or you can say stuttering transcription. Now last two steps, that is assembly and release of virion. Generally, assembly take place inside the cytoplasma, and that self-assembly will produce the nucleocapsid. So nucleocapsid, while production of nucleocapsid, N protein, L protein and phosphoprotein will come, that will produce ribonucleoprotein complex, and that ribonucleoprotein complex, under the influence of matrix protein, that will align at the cell membrane where glycoproteins are inserted. Now this from this point they will comes out by budding, and they will release in environment, and that will infect the new cell. This is in brief about the replication of rhabdoviruses. Now in specific, if you will see, these are the three important viruses, namely vesicular stomatitis virus, rabies virus and ephemeral fever virus. So vesicular stomatitis generally considered as cytopathology. It means they are the rapid grower and they can destroy the cell. In contrast to this virus, rabies virus and bovine ephemeral fever virus, they are considered as a non-cytopathic, they are slow in growth, and they does not kill the host cell, because they simultaneously allow the cell protein production and nucleic acid synthesis. Now the last characteristic of rabies virus, that it produces Negri body. This is an intracytoplasmic inclusion body, that is a characteristic of Negri, that is a characteristic of rabies disease, and it helps in diagnosis of rabies disease. Generally, cellular stain is used for diagnosis or demonstration of Negri body, and it is named based on the scientist name, and presence of Negri body invariably tells that the sample is positive for rabies. But absence of Negri body in the suspected tissue is never conclude that sample is negative for rabies virus. That always demands second test for confirmation of true negative sample. The last point, that is a DI particle. In general virology we had discussed about DI particle. Generally this is a defective interfering particles, or you can say they are the deletion mutant. They are truncated, that is truncated genome, and because of truncated genome, generally that favors or that has upper hand in replication over the normal virus. So that's why whenever we are cultivating viruses in vitro system like the cell culture, if you are going for high MOI, that is multiplicity of infection, that will leads to the production of high amount of DI particle. And this is a characteristic of many other viruses too. So this is in brief about the classification, virion property, genome property and replication of rhabdoviruses. Now to to summarize this replication cycle, this is a schematic diagram which tells that this is a virus, enveloped virus. This represents the rhabdovirus that will attach to the specific receptor present over the cell membrane. Once it will attach, that will get internalized in these vacuoles. These endoplasmic vacuoles, their membrane get fused with envelope, and that process is pH dependent. Once it will get fused, that will leads to the uncoating of nucleocapsid, and uncoating of this ribonucleoprotein leads to exposure of RNA genome for transcription and translation process. In transcription, that will produce 5 messenger RNA, and this 5 messenger RNA will produce corresponding protein by translation. The first glycoprotein that undergo various post-translation modification, and that will comes or studied on the cell membrane where budding will happen. M, that is a matrix protein that will comes or that will helps for assembly of virus, and P and L protein, they are generally associated with genome, and that genome will produce the RNP complex, and after production of RNP complex, that will comes or align at this particular place where already G proteins or G genes are studied, and once it will align, it will produce or it will take up the envelope and it will be bud out from the cell. So this is a brief about the rhabdovirus replication. Now to summarize this class, there are 4 important points you have to remember. One, this particular family follow under mononega virus, that is Rhabdoviridae, then that has 3 important genus and 3 important viruses. These viruses, they have peculiar shape that is known as bullet shape, and generally they are enveloped one. In case of genome, that is single molecule, linear, negative sense, single stranded RNA. This contains 5 important genes, and that 5 important genes are present in this fashion from 3' end to 5' end, and replication process or transcription process will follow attenuation transcription. That is also known as top start transcription or stuttering transcription. And at the last, generally this replication of this virus takes place in cytoplasma, and it produces the Negri body, which is a characteristic of rabies virus, and sometimes it also produces DI particle. So this is all about the general aspect of Rhabdoviridae. In next class, we will study in detail about the various important diseases like rabies, vesicular stomatitis, and bovine ephemeral fever virus. So if you have any doubt, you can call me, you can ask me, and your suggestions are always welcome. Thank you once again for listening me.